Buenas tardes. Hola. Vamos a dar comienzo a esta conferencia. Vamos con retraso. Os pediría, por favor, los que estáis abandonando la sala, que lo hagáis en el máximo silencio porque vamos muy atrasados. La doctora Ávila me ha pedido excusas por acabar tarde. Me ha comentado que también ellos han empezado tarde. Y bueno, gracias a los que os estáis quedando a la hora de comer para asistir a esta conferencia. La conferencia, sabéis que es sobre avances en el conocimiento de la fisiopatología del fallo de membrana peritoneal. Yo creo que el tema, para los que nos gusta la diálisis peritoneal, es un tema apasionante. Todos sabemos que el fallo técnico es uno de los problemas que tiene la diálisis peritoneal y que el fallo de membrana está metido en esa ecuación del fallo técnico. Sabéis que tenemos buenas noticias al respecto, tenemos menos esclerosis peritoneal de la que teníamos hace unos años, tenemos mejores soluciones, tenemos un mejor conocimiento fisiopatológico de las acuaporinas, etcétera, pero bueno, todavía queda mucho camino que recorrer. Y para hablarnos de esto tenemos al doctor Johan Morell. El doctor Johan Morell a mí me es una alegría tremenda tenerlo aquí, porque intentamos tenerlo en la Reunión Nacional de Diálisis Peritoneal y Hemodomiciliaria de Albacete, pero por problemas de agenda no pudo venir. Finalmente hemos conseguido traerlo. El doctor Morell tiene una experiencia tremenda en el campo que nos ocupa, tiene publicaciones como primer autor hechas en Kidney International, New England y en un montón de revistas, y es el primer firmante de la declaración de la Sociedad Internacional de Diálisis Peritoneal sobre cómo estudiar, abordar y tratar el fallo de membrana. Por tanto, es una auténtica autoridad en el tema y va a ser un placer escucharle. Dr. Morell, thank you very much for coming, and please feel free to start whenever you want. Thank you.
Muchas gracias. I would just say a few words in Spanish for the introduction. So, estimados colegas y amigos, es un tremendo honor y placer dar a esta conferencia hoy y agradezco al comité organizador por la amable invitación para hablar sobre los mecanismos de disfunción de membrana en diálisis peritoneal. Esta es mi declaración de conflicto de interés. Como sabéis, la diálisis peritoneal o DP ofrece varias ventajas para tratar la insuficiencia renal. Es altamente efectiva y segura, por lo que representa la principal modalidad de diálisis domiciliaria en todo el mundo y promueve la autonomía de los pacientes, lo que por supuesto es muy importante para los pacientes con enfermedades crónicas. Además, la DP también ayuda a preservar la función renal residual, reduce la exposición al COVID y es menos costosa que la hemodiálisis en el centro. Sin embargo, la diálisis peritoneal también tiene algunos inconvenientes, incluida la sobrecarga de líquidos, que sigue siendo muy común, con más del 50% de los pacientes prevalentes en DP con hipervolemia. Es importante destacar que la presencia de hipervolemia se asocia con un mayor riesgo de muerte y otras complicaciones. Como resultado, la eliminación de agua o ultrafiltración peritoneal es clave para restablecer el equilibrio de líquidos y la eficiencia de la diálisis peritoneal. Los objetivos de esta lección son entender cómo funciona el peritoneo como membrana de diálisis, identificar los mecanismos de disfunción de la membrana y destacar el papel de la genética y la fisiología de la membrana. Now I will switch to English, and this is a pathological section of the peritoneal membrane. You can see that it contains 3 layers, mesothelial cells, the peritoneal interstitium, and a dense network of microvessels. At the level of the microvascular endothelium, there are 3 types of pores that were predicted to explain the transperitoneal movement of water and solutes during peritoneal dialysis, and among these 3 pores, abundant small pores allow the diffusion of small solutes like creatinine and urea and the absorption of glucose as well as the movement of water coupled with sodium. The ultra-small pores were predicted to be located at the level of endothelial cells and to mediate the movement of water without solutes, also called free water transport. Approximately at the same time the 3-pore model for peritoneal transport was postulated, Peter Agray and his team in the USA identified very small proteins with properties of water channels on the surface of red blood cells. As these unknown proteins facilitate the diffusion of water molecules across biological membranes following an osmotic gradient, Peter Agray called them aquaporins. These aquaporins were initially found to be expressed in red blood cells and in kidney tubules, but they are also present in the peritoneal membrane, as illustrated here using immunohistochemistry and electron microscopy, where you can see that all these small black dots are actually aquaporins in the membrane of endothelial cells. The development of a mouse model of PD and its application to transgenic mice lacking water channels demonstrated the important role of aquaporins in peritoneal ultrafiltration. The instillation of hypertonic glucose into the peritoneal cavity of wild-type normal mice in red drives ultrafiltration and increases the intraperitoneal volume over time, and it also generates the sieving of the dialysate sodium, which is the rapid fall in dialysate sodium concentration during the first part of the dwell. The deletion of the aquaporin-1 gene in mice decreases the permeability of the membrane to water, reduces total ultrafiltration by approximately 40 to 50%, and completely abolishes the sodium sieving, demonstrating that the aquaporins are actually the molecular counterparts of the predicted ultra-small pores. So based on this brief reminder of peritoneal physiology, we as clinicians need three types of information to evaluate the peritoneal membrane function in our patients. First, we want to know about the diffusion rate of small solutes. We want to quantify the total water transport, also called ultrafiltration, and we would like to estimate free water transport across the aquaporins. To achieve these goals, Professor Verger in France and Professor Twardowski in the US proposed the peritoneal equilibration test more than 30 years ago. The peritoneal equilibration test, or PET, consists in a standardized forward well using glucose-based PD solution to quantify the equilibration rate of small solutes across the peritoneal membrane. This procedure was shown to be safe, easy, and accurate, as well as highly reproducible. And later, a modified version using hypertonic glucose was proposed to quantify water transport more accurately and to assess the sodium sieving. Evaluating membrane function in PD is very important because there is, as you can see on this slide, a huge variability in membrane characteristics between the patients already at PD start, and this is true for peritoneal solute transfer rates on the left, net ultrafiltration, and sodium sieving. In addition, this variability associates with outcome, as we will see in the next couple of slides, and has a clinical impact on fluid removal and dialysis prescription. This was recently reviewed and discussed in the ISPD guidelines for the evaluation of membrane function and dysfunction in PD patients, and I propose here to summarize the key messages of these recommendations. The definition of membrane function highlights the importance of ultrafiltration and fluid balance in PD patients, and membrane dysfunction is therefore defined as a membrane that fails to achieve sufficient ultrafiltration to maintain adequate fluid status and or by a low ultrafiltration capacity during the PET. Of course, in the absence of any catheter dysfunction or mechanical problems. Second, based on the underlying pathophysiology and clinical observation, membrane dysfunction can be classified into three different causes. First, fast peritoneal solute transfer rate, where the rapid diffusion of small solutes will cause fast glucose absorption and dissipation of the osmotic gradient with entering loss of ultrafiltration capacity. Second, acquired membrane insufficiency, when the membrane becomes less effective over time on PD. And lastly, poor intrinsic ultrafiltration, meaning that the membrane is poorly permeable to water from the start of PD. Fast peritoneal solute transfer rate is defined as a high dialysate over plasma creatinine ratio at the end of the PET. PSTR is influenced by different determinants, and the most important ones are intraperitoneal inflammation with IL-6 levels, as well as genetic variation. The importance of genetic variation on peritoneal solute transfer rate was recently confirmed by the BioPD study led by Raj Mehrotra in collaboration with PDOPS and many others throughout the world. And the BioPD study was actually the first genome-wide association study in peritoneal dialysis, and as such, it looked for potential associations between gene variants along the entire genome and baseline peritoneal solute transfer rate. The study included almost 3,000 incident PD patients, and it showed that the importance of genetics or genetic factors explain approximately 20% of the variability in solute transfer rate at the start of PD. The BioPD study also showed that polygenic risk score associated with solute transfer rate, and it identified 4 loci with suggestive associations with solute transfer across the membrane, as illustrated by this Manhattan plot. Fast PSTR has huge clinical implications and has been associated with a higher risk for death in patients in this meta-analysis released in 2006, mainly including patients treated with continuous ambulatory PD and receiving glucose as only osmotic agent. And this, the importance of fast PSTR on outcome has been confirmed in a more recent and more contemporary cohort, including more than 10,000 patients from the US, and fast PSTR at the start of PD was again shown to be associated with all-cause mortality, technique failure, and the risk of hospitalization. The higher risk in patients with fast PSTR at baseline is at least partly due to the rapid diffusion, rapid absorption of glucose. Glucose is indeed a very small molecule, rapidly absorbed across the peritoneal membrane, with a transient effect on ultrafiltration, especially in patients with a fast transport status who have a large number of small pores. In contrast to glucose, icodextrin is a mixture of glucose polymers, which include also large colloidal fractions. It induces a slower but more sustained ultrafiltration than glucose, and in clinical trials, icodextrin was consistently shown to improve technique survival, metabolic control, and net ultrafiltration, especially in patients with fast PSTR at baseline. And so in patients, based on these underlying mechanisms, it is advised to shorten the duration of glucose-based dwells and to use icodextrin in patients with fast PSTR. And therefore, membrane testing offers an opportunity for intervention, and several studies have shown that adaptation of PD prescription can improve the outcome and reduce mortality in patients with fast PSTR, as in this study by Simon Davies from the Stoke cohort. The second type of membrane dysfunction is acquired membrane insufficiency. Indeed, the peritoneal membrane is a living tissue that undergoes structural and functional changes over time on PD, and you can see that there is a progressive fibrosis, vascular proliferation, faster solute transfer rates over time on PD in orange, and answering loss of ultrafiltration capacity. Encapsulating peritoneal sclerosis is, although very rare, the most dramatic complication of long-term PD. It is characterized by an excessive fibrogenic response of the membrane, as illustrated here, that will ultimately encapsulate the bowel, leading to intestinal obstruction. Several registry and cohort studies have consistently shown that peritoneal transport defects, including acquired and progressive loss of ultrafiltration capacity and loss of sodium sieving, precede and even predict the development of overt EPS, and these transport defects were linked to specific changes in the peritoneal interstitium of patients with EPS. And you can see that the peritoneal membrane of those patients on the right in red is particularly thickened and dense in collagen fibers. And so, as we don't have any specific predictor for the development of EPS, it has been suggested, based on these data, that monitoring water transport during time on PD can help identifying patients at risk for EPS, who might have a disproportionate loss of water transport over time on PD. The third type of membrane dysfunction is intrinsic low ultrafiltration, which means that the patient have a low net ultrafiltration capacity and a low sodium sieving from the start of PD. You can see on this slide that there is a huge variability in water transport from the start on PD, and unfortunately the causes of this variability were not very well understood. So as water transport is a critical determinant of the efficiency of PD, as aquaporine water channels mediate half the ultrafiltration in PD using glucose, And as the huge variability in water transport remained poorly understood, we asked the question, could there be a role for genetic variation in AQP1, the gene encoding for water channels, to explain this variability in water transport and to investigate the potential association between aquaporin-1 gene variant and peritoneal water transport. We gathered clinical data and DNA from almost 2,000 patients treated with PD from 7 cohorts in Europe and Asia, and we genotyped those patients for common variants in the aquaporin-1 gene, covering the major linkage disequilibrium block over the small AQP1 gene. First, in a discovery phase, we confirmed the huge variability in net ultrafiltration and sodium sieving at the start of PD. During the peritoneal equilibration test performed at baseline. And then we looked for potential associations between several gene variants and water transport. And we identified one specific variant, the RS2075574, which was associated with defective water transport. Indeed, patients carrying 2 copies of the variant, so with a TT genotype, represented approximately 10 to 15% of the cohort, and those patients had a lower net ultrafiltration and lower sodium sieving as compared to the patients with a CC genotype, although the solute transfer rate was similar. In a validation phase, we confirmed the association between the variant and daily net ultrafiltration, which is the total amount of water removed across the peritoneal membrane during a day. And this parameter, of course, has high clinical relevance. And again, patients with a TT genotype in red had a lower daily net ultrafiltration of approximately 200 mL as compared with patients carrying the 2 ancestral C alleles in gray. And again, this was independent from other characteristics, including peritoneal solute transfer rate. Next, we investigated the potential relevance, biological relevance of this variant. Interestingly, the variant we identified is located in a highly conserved region of the aquaporin-1 promoter, where it is known to regulate the expression of erythroid-specific genes. Using the luciferase reporter assay in vitro, we showed that the T allele in red decreased the activity of the promoter as compared with the C allele in gray, meaning that there is less, there is a reduced activity of the promoter and less transcription of the AQP1 gene. Next, using peritoneal membrane collected at catheter insertion, we observed decreased levels of mRNA and protein levels of AQP1, which is here stained in green in the peritoneal membrane of patients with a TT genotype as compared to those with the CC genotype. And the lower expression of water channels was also observed in the membrane of red blood cells of patients with a TT genotype as compared to those with a CC genotype. And this was associated ex vivo with a reduced permeability to water, but not to solutes of those red blood cells membranes. So a common variant in the aquaporin-1 gene with a functional relevance is associated with defective water transport across the peritoneal membrane. And as water transport is a critical parameter of PD efficiency and outcome, the next question was, does the variant associate with outcome in incident PD patients? Among the almost 900 patients included in the outcome analysis, those carrying 2 copies of the risk variant in red at a shorter time to a composite of death or technique failures compared to other patients. And you can see that the association between the variant and outcome was consistent across all subgroups and mainly driven by a higher all-cause mortality associated with the TT genotype. So the last question we asked, is it possible to mitigate poor outcomes associated with the aquaporin-1 risk variant? In a previous work that we performed in experimental models, we investigated the mechanisms of crystalloid and colloid osmosis across the peritoneal membrane. We knew already that the glucose relies on the presence of aquaporin-1 water channels to drive ultrafiltration, but the potential role of aquaporine water channels in icodextrin-driven ultrafiltration was unknown. And using a combination of aquaporine-1 null mice, biophysical studies, and computer modeling, we showed that icodextrin actually induces a water transport that is completely independent from the presence or not of aquaporins. And more recently, we observed that icodextrin induces a water transport that is unchanged both in heterozygous aquaporine mice or in patients carrying the deleterious TT genotype, in which there is an approximately 40 to 50% reduction of the expression of water channels in the peritoneal membrane. So this large collaborative effort was allowed thanks to the participation of many centers, including in Spain, including Rafa Selgas and Maria Barro from Madrid and their collaborators. And this work was the first to demonstrate the influence of genetic factors on PD efficiency. Using a combination of genetic, translational, and clinical data, we identified an aquaporine-1 promoter variant that influences water transport and is associated with outcome in PD. This risk variant has a biological relevance as it influences the activity of the aquaporine-1 promoter and the expression of water channels in the peritoneal membrane. Preliminary studies suggested that colloid osmotic agents may mitigate the risk associated with the variant, but this will need to be confirmed in prospective trials. So to conclude, I will say a few words again in Spanish and in my notes, sorry. Para concluir esta conferencia, podemos decir que restaurar y mantener el equilibrio de líquidos es fundamental para el éxito del ADP. Comprender cómo funciona el peritoneo como membrana de diálisis es clave para individualizar la prescripción y mejorar el resultado clínico. La disfunción de la membrana contribuye a la sobrecarga de líquidos en la ADP. Por último, una variante del gen AQP1 influye en el transporte de agua y el resultado en la ADP. Es por esto que el estudio de la membrana y posiblemente la detección genética pueden considerarse como una forma de medicina de precisión en diálisis, teniendo en cuenta la variabilidad para individualizar la prescripción y así mejorar los resultados en nuestros pacientes. Quisiera agradecer a los pacientes y enfermeras de las unidades de diálisis domiciliaria, los compañeros que contribuyeron a estos estudios en Bélgica, España, Europa y Asia. Muchas gracias por su atención.
Muchas gracias, doctor Morell, por una magnífica presentación. No sabía que hablaba tan bien español. No sé si querrá las preguntas en español o en inglés. Would you prefer questions?
Oh, maybe questions in English, please.
Okay. Vamos a ver un pequeño turno de preguntas. Sabéis que tenemos poquito tiempo, pero bueno, yo creo que hay algunas preguntas sí que tenemos que cuestionarlas. Los que queráis, por favor, hacer alguna. ¿Sí?
Sí. La tengo que hacer en español.
La pregunta la puedes hacer en español y la puedo traducir yo, si no te manejas en inglés, como quieras.
Bueno, enhorabuena. Nos ha encantado la presentación y todos los trabajos que lleva haciendo. Mi pregunta es, con toda la evidencia que hay de lo importante que es medir el transporte de agua, no podemos hacer estudios genéticos todos, pero sí que podemos hacer cinéticas con glucosa hipertónica. Y las podríamos hacer desde el inicio y las podríamos hacer en la evolución. A mí me sorprende que en las guías ISPD que han publicado y que es el principal autor, se siga sin recomendar la cinética al 386 con medida del cribado de sodio. Nos ayudaría a nivel basal para saber si puede tener un déficit de aquaporina. No tenemos estudios genéticos y nos ayudaría en anticiparnos a la esclerosis peritoneal mucho más que con las cinéticas que estamos haciendo. No sé si me puedo traducir, si me ha entendido.
Lo puedo intentar, a ver lo que... Did you understand that?
I'm sorry, no. Not
Okay,
everything I
she wants to know why, if aquaporine transport is so important, and we already know that, why the ISPD guidelines are not including the use of this PET test with 4.25% as a tool, as a daily tool, well, not daily, but monthly or yearly tool to study the evolution of the transport in our patients. Why it's not recommended as a usual way of work?
Yeah, so thank you very much for the question. I will answer, of course, in English. But this was a long discussion, actually, in the working group of the ISPD for the recommendations. Should we recommend using hypertonic glucose or not? We know currently that, so we don't have lots of studies to support the use of hypertonic glucose instead of intermediate tonicity glucose, 2.5%. So we lack studies first. We know that many centers do not perform tests at all. And if they do, most of them currently use the intermediate tonicity glucose solution, so 2.5% dextrose. So we don't want to change everything, and we know also that the most evaluated parameter is peritoneal solute transfer rate so far. We have lots of studies about the D over P creatine ratio. We have very few studies about the impact of water transport on long-term outcome, and we need more studies to support the systematic use of hypertonic glucose.
Mm-hmm. No sé si lo has entendido.
Sí.
Vale, perfecto. ¿Tiempo para una última pregunta?
Hola, estoy aquí.
Sí, adelante, Miguel.
Doctor Morel, muchas gracias por su magnífica conferencia. Yo quería preguntarle, los pacientes que tienen una variante perjudicial con baja expresión de aquaporina tienen una mayor mortalidad. ¿Esto es debido a un fenómeno local de baja ultrafiltración o puede haber otros efectos perjudiciales en otras partes del cuerpo de la baja expresión de aquaporina?
Do you understand?
I think I understood. So is it only the role of defective water transport that explains the higher risk of mortality, or may we postulate the existence of other factors? Is it correct?
Yeah, the idea is if this failure in aquaporine genetic with higher mortality, is it due to peritoneal effects, or is it due to effects on any other endothelium or?
Yeah, so that's a very good question, and we don't have the final answer. Of course, aquaporins are very important for water transport, and there is an effect on ultrafiltration, that's for sure. We know that aquaporins, however, are expressed in a very large number of cells throughout the organisms, including inflammatory immune cells, and we cannot rule out a potential impact of the genetic variant on those other cells. So we were to be honest, we were very impressed to see the higher mortality in patients with carrying the 2 copies of the variant. And so far, I cannot answer. I cannot provide any definitive answer, and this will require future works to identify the potential roles of inflammation. We know that aquaporins are also important in the heart, for instance, in the vessels. So there might be pleiotropic effects of aquaporins across the organism.
Bueno, pues dejamos aquí con más retraso debido a la conferencia. Muchísimas gracias por haberos quedado hasta esta hora y demostrar la pasión que tenéis por este tema. Y muchísimas gracias, por supuesto, al doctor Morel. Thank you very much, Dr. Morel.
Muchas gracias.